Welcome back to MLT 240. This is the last part of the mycology lecture. In this portion of the lecture, we're going to be covering subcutaneous mucosis. Some general characteristics of subcutaneous mucosa are that these are fungal diseases that affect the subcutaneous tissues that usually result from traumatic implantation of foreign objects into deep layers of the skin. So that's how the fungus gains entry to the host. So for example, through rose thorns, through the soil, through punctures in the skin. The primary site of infection a lot of times is the feet. These fungi are commonly found on the soil or decaying vegetation, so agriculture workers are most often affected. Infections are commonly chronic and usually incite the development of non-healing, hard, lumpy, or crusted lesions at the site of the trauma. Infections rarely disseminate or spread to other areas of the body. Subcutaneous mycosa are often caused by diatomaceous molds, hyaline molds, or fungus-like bacteria. Subcutaneous mucosa may be classified by the disease process they cause or by the causative agents that are involved. Here are some of the subcutaneous mycosis that we are going to discuss. Chromoblastomycosis, eumycotic mycetoma, subcutaneous phaeohyphomycosis, and attendomycosis. Chromoblastomycosis is also known as verrucous dermatitis, and it causes types of skin lesions, and it's also known as chromomycosis. Chromoblastomycosis causes chronic skin infections of the skin and subcutaneous tissues, and it develops over months and years. It causes lesions usually confined to the extremities, such as the feet and lower legs. It appears as verrucous nodules that become ulcerated and crusted. They're long-lasting lesions that have a cauliflower-like surface. Sclerotic bodies or black dots are deep in the part of the lesion, and those are diagnostic for this disease. This disease occurs worldwide, most commonly in the tropic or subtropic regions of the Americas and Africa. In the U.S., most cases are in Texas and Louisiana. And we'll see examples of this fungi in just a minute. There are several infectious agents in this category that can cause chloroblastomycosis. There's Phonosecia that can cause diatomaceous branching conidia. There is Phylophora, which is also diatomaceous, and it forms balls of conidia. Next is Cladophylloporia, which is a diatomaceous branching conidia. And finally, there's Rhinocladilla, and this is a fungus where conidia are only on the upper portion of the conidiophore. Here are some images of the lesions that are caused by chromoblastomycosis. You can see those ulcerated, crusted, long-lasting lesions, and of course some of them have that cauliflower appearance. Next up is eumycotic mycetomas. These are chronic granulomatous infections of subcutaneous and cutaneous tissues, and they arise at the site of inoculation. So wherever the skin is pricked or punctured is where the infection happens. It's characterized by swelling with exudate draining of the skin surface through sinus tracts. Mycetomas can be caused by a fungus or a bacteria. These infections usually occur primarily in tropical and subtropical areas, but also can be seen in temperate zones, and these fungus usually are endemic in areas such as India, Africa, and South America. A couple of the causative agents are Pseudosporum and Marley-Trotter, but Marley-Trotter causes most of the cases. Next is phaeohyphomycosis. It's caused by dark pigmented fungi, and the fungi that have melanin in their cell walls. They may form yeast-like cells that are solitary or in short chains, or hyphae that are septate, branched, or unbranched. and are often swollen or irregular beaded in appearance. Causative organisms are commonly found in nature and encompass many genres. Species regularly associated with this condition include Phylophora, which produces balls of conidia. Next is actinomycosis. This is a chronic granulomatosis infectious disease characterized by the development of sinus tracts and a fistula which erupts to the surface and drains pus containing sulfur granules. Actinomycosis can be caused by Actinomyces israeli and other related organisms. Another species that can cause subcutaneous mucosa is Sporothrix syncytia. It causes lymphocutaneous sporothrotosis, which is a chronic infection characterized by nodular and ulcerative lesions among the lymph channels that drain primarily at the site of inoculation. Members of this complex are commonly recovered from the soil and decaying vegetation, and distribution is worldwide. It's found in temperate countries such as France, Canada, and the U.S. Most cases are associated with guarding, especially with exposure to rose thorns. It's also known as rose handler's disease. This species is known to be dimorphic, with mold forming at 25 to 30 degrees Celsius and giant cigar-shaped yeast at 37 degrees Celsius, which is body temperature. You usually have fruitiform or teardrop-shaped microconidia in a daisy head pattern. Here is an example of the microconidia, and it being teardrop shaped and it forming a daisy head pattern for rose handler's disease. And you can see several of these are kind of in a daisy head pattern, and the microconidia are teardrop shaped. Here is a list of fungus-like bacteria. First off is Nocardia. It's partially acid-fast as far as staining. It can be confused with tuberculosis. It causes pulmonary and cutaneous infections, and it has a very high mortality rate. Next is Streptomyces. It can cause actinomycotic mycetoma. Then there is Microbacteria, which can cause TB or tuberculosis. There's Actinomyces israeli. If in the oral cavity, it can cause lumpy jaw, but if it gets into the lungs, it can cause farmer's lung. Now we're moving on to cover our systemic mycosis. Here are some general characteristics of systemic mycosis. These are generally diseases contracted through inhalation of infectious conidia, and they disseminate throughout the body's organs and systems. But any fungus in an immunocompromised host has the potential of becoming systemic. The organisms in this category, the systemic mucosa, are dimorphic, meaning they have a mold form and a yeast form. Some of the causative agents for systemic mycosis that we're going to discuss include blastomyces, coccidioides, histoplasma, paracoccidioides, Taylormyces. Our first systemic mycosis is blastomyces dermatitis. It's most common in middle-aged men. Patients usually exhibit flu-like symptoms and may eventually lead to a progressive pulmonary or invasive disease. There's ulcerative lesions in the skin and bone. It's endemic to the Mississippi and Ohio River basins, and also it occurs in Africa. There's a narrow range of conditions that appear to support growth. In patients, you can usually examine tissue or purulent or pus-like discharge in cutaneous skin lesions. The yeast cells are usually large spherical cells with double contoured thick walls, and they have buds that connect by a broad base. In the mold form, the conidia are on short lateral branches and are ovoid to dumbbell-shaped and resemble a variety of other fungi. Here are a couple images of the yeast form of blastomyces. You can see the double-based budding and double-contoured walls of the yeast form. The next species is Coccidioides immitis. This species is probably the most virulent or most severe or harmful of all the human mycotic agents. Inhalation of just only a few arthrocanidia can produce primary coccidioides mycosis. It's also known as valley fever or San Juan Valley fever or desert rheumatism. Laboratory workers have to be really cautious when working with suspected samples. It can lead to progressive pulmonary disease or multi-symptom dissemination, or it can spread throughout the body. Filipino populations and black men are at high risk for dissemination. This species is endemic in the desert areas of the southwest U.S. It's very resistant to heat, dryness, and salinity, or salt and water. Upon tissue examination, you should be able to see giant spicules containing endospores. During culture examination, you should see fertile hyphae arising at right angles to the vegetative hyphae, producing alternating hyaline arthrocanidia and barrel-shaped arthrospores. As far as microscopic ID, this is a tissue sample, and you can see the spicules containing endospores. This is another microscopic image, but this is in the mold phase, and you're able to see the barrel-shaped arthrospores. Next up is Histoplasma capsulatum. This causes histoplasmosis. Some other names for this disease include reticuloendothelial cytomycosis, cave disease, and spelunker's disease. Spelunkers are referring to cave explorers. This disease is acquired by inhalation of microcanidia. The microcanidia are phagocytized by macrophages in the pulmonary parenchyma, so this affects the RE system, or the reticuloendothelial system. Most patients are asymptomatic, but in immunocompromised patients, it can cause progressive and potentially fatal disseminated disease. In the U.S., the endemic areas are Ohio, Mississippi, and the Missouri River deltas. The organism resides in soil with high nitrogen content, especially areas contaminated with bat and bird guano. And guano is just another word for bat and bird poop. You'll see small yeast cells in direct smears. A lot of times our stain smears will often show yeast with monocytes and macrophages. Here are some images of histoplasma. The first one is an image of histoplasma on a culture plate at 30 degrees Celsius. It's a white cottony colony. The second image is of the organism at the tissue phase. And the last one at the bottom is histoplasma in the mold phase. This is an image of histoplasma in a blood sample. You can see the arrow pointing to the actual organism for histoplasma. The next species of systemic mycosis is Paracoccidioides brucellensis. It causes South American blastomycosis. It's a pulmonary infection, usually asymptomatic, though dissemination can lead to ulcerative granulomatous lesions of the buccal, nasal, and GI tracts. This species has a narrow range of temperature tolerance. It prefers cooler areas of the body. Geographic areas of higher incidence require humid, high rainfall areas with acid soil concentrations. So it usually is around areas such as South and Central America. For microscopic review, we can use direct microscopic examination of cutaneous and mucosal lesions. We'll typically see budding yeast cells with multipolar budding at the periphery, resembling a mariner's wheel, and we'll see that here in just a minute. When there's only a few buds, it's said to look more like a Mickey Mouse head, or it's called Mickey Mouse. The mold produces small one-celled conidia. Here is a microscopic image of Paracoccidioides, and this is supposed to resemble the mariner's wheel or a ship's wheel. So this is the center, and these would be like the spokes or the grips on the mariner's wheel. The last systemic mycosis in this section is Ptoloromyces morneffi. It's the only true pathogen in this genus. It's a common cause of systemic infection in immunocompromised patients who have visited endemic areas of Southeast Asia. Infections usually disseminate with multiple organ involvement and is typically fatal. The fungus can be isolated from cutaneous lesions. Oval to cylindrical yeast-like cells can be detected in right-stained smears from skin lesions or biopsy specimens. The mold form has sparse green aerial and reddish-brown vegetative hyphae and produce a red diffusible pigment. The last two slides are on some different drugs that we use to treat fungal infections. The first drug and most popular is the amphotericin or amphotericin B or AMB. It's toxic to the fungi and also to patients. It can lead to renal impairment in patients, but it remains the drug of choice for most life-threatening fungal diseases. But we are seeing some resistance beginning to emerge to this drug. Next is fluconazole or intraconazole. These are reasonably active against fungi and with fewer side effects. Fluconazole is the leading agent for yeast infections, but limited to no activity against molds. But the overuse or misuse has resulted in some development of resistance to these drugs. Some of the other azoles are usually more frequently prescribed for treating mold infections. Caspofungin is lethal for yeast. It's very effective for aspergillus. Good option for resistant fungi. It targets the cell wall synthesis. Terbinafine and naptiphyne is in the last class of drugs given for fungal infections. They're usually given orally and activate against several groups of fungi, but naptiphyne is usually given topically. This concludes our final portion of our mycology lectures. If you have any questions or need further clarification, please don't hesitate to reach out, and I will see you over in our next lecture.